This kills stage 4, cancer cells, how to starve and prevent colon cancer. The alarming increase in colon cancer diagnoses among individuals in their 20s and 30s, a stark contrast to when it was once a rarity before age 50, doesn't stem from our genes. Instead, the primary drivers are found in what we consume and what's notably absent from our diets. Many people hold the misconception that a diagnosis of stage 4 cancer signifies an irreversible end, believing that once cancer spreads, it's a death sentence, and that our food choices have minimal bearing on the outcome. However, after dedicating nearly 15 years to emergency medicine and closely collaborating with oncology patients, I've observed firsthand phenomena that challenge established medical teachings. It's astonishing to realize that advanced stage 4 colon cancer can potentially be reversed to stage 0, leveraging the body's own immune system. Furthermore, the foods we incorporate into our meals possess the remarkable ability to starve tumors by severing their blood supply. A cancer that cannot establish new blood vessels is severely limited. It cannot grow beyond the size of a ballpoint pen's tip. Conversely, if a tumor gains access to a blood supply, it can experience explosive growth, expanding to an astonishing 16,000 times its original size in a mere 14 days. I am Dr. Becker, an emergency medicine physician, witnessing an ever-increasing number of younger patients diagnosed with colon cancer. Individuals who should have many decades of healthy life ahead of them has been deeply troubling. This surge in colon cancer among young adults is not genetically predetermined. It's an environmental issue directly linked to our eating habits and nutritional deficiencies. In this discussion, I will comprehensively explain how to activate your body's inherent defenses to combat cancer and prevent colon cancer, and how individuals with advanced disease are achieving complete remission. We will delve into the reasons behind the skyrocketing colon cancer rates in younger populations, explore the groundbreaking immunotherapy treatments that can transform stage 4 into stage 0, and uncover how the composition of your gut bacteria plays a crucial role in the success or failure of cancer therapies. Additionally, we will identify specific foods that can effectively starve tumors by hindering their ability to develop new blood vessels. Most importantly, I will present a precise dietary protocol that you can implement starting today, with the potential to reduce your colon cancer risk by as much as 40%. Before we proceed, I would greatly appreciate a small favor. I've noticed messages from individuals who have benefited from watching multiple videos, but may have forgotten to subscribe. Could you take a moment to double-check your subscription status right now? It's entirely free, takes mere seconds, and significantly helps this channel reach more people seeking authentic, science-based health information. Let's begin with a startling observation. 20 years ago, during my medical school years, a diagnosis of colon cancer in someone under 50 was so unusual that it warranted presentation at grand rounds. Today, however, I am encountering patients in their 30s, 20s, and even teenagers who are diagnosed with colorectal cancer. The incidence of this disease in individuals under 50 has seen an approximate 50% increase over the last 3 decades. This indicates a profound alteration in our environment, with compelling evidence overwhelmingly pointing to diet and lifestyle as the primary culprits. It's important to understand that only a small fraction of cancers, perhaps 10–15%, are primarily driven by inherited genetic mutations. The overwhelming majority are the result of environmental factors interacting with our biological makeup. So, what exactly has changed? We are consuming more processed foods than at any other point in human history. Our diets are laden with massive quantities of processed meats, including hot dogs, deli meats, bacon, and sausage, all of which the World Health Organization has classified as Group 1 carcinogens, placing them in the same category as cigarettes. These foods don't merely contribute to a slight increase in cancer risk. heavy consumption of processed meats is associated with an 18% elevated risk of colorectal cancer for every 50 grams consumed daily, which is roughly equivalent to 2 slices of deli meat. Consider the journey of processed meat through your digestive system. It resides in your colon for hours before elimination. During this time, compounds present in the meat, such as nitrosamines, heterocyclic amines, and hemiiron, create an inflammatory environment that damages the cells lining your colon. Chronic inflammation is a fundamental precursor to cancer development. Your cells are in a constant state of repair, and when this repair process is overwhelmed, mutations begin to accumulate. However, diet is not the sole factor. A sedentary lifestyle represents a significant risk factor. When you spend your day without physical activity, your metabolism slows, inflammation increases, your gut bacteria become unhealthy, and your immune system weakens. Engaging in regular movement, even a brisk 30-minute walk daily, enhances gut motility, reduces inflammation, and strengthens immune surveillance. Furthermore, smoking and vaping are detrimental. Many mistakenly believe vaping is a safer alternative to cigarettes. This is incorrect. The chemicals found in vaping liquids can be even more toxic than tobacco smoke in certain respects, and they unequivocally increase the risk of colorectal cancer. Alcohol consumption is another major contributor. Even moderate alcohol intake elevates colon cancer risk through various mechanisms, including DNA damage and disruption of folate metabolism. What unites all these risk factors is their ability to suppress your immune system. Your immune system is one of the body's 5 critical health defense mechanisms and is arguably the most vital for cancer prevention. Imagine your immune system as a vigilant police force constantly patrolling your body on the lookout for threats. You possess specialized immune cells, known as natural killer cells and T cells, which function as elite soldiers expertly trained to identify and eliminate abnormal cells, including cancerous ones. Right now, as you are reading this, microscopic clusters of cancer cells are likely forming within your body. This is a normal biological process. With trillions of cells undergoing constant division, errors inevitably occur. Some of these errors lead to the formation of cells that are technically cancerous. However, your immune system is adept at detecting and neutralizing these cells before they can pose a serious threat. It's akin to having security cameras and armed guards protecting a building. Most threats are neutralized before causing any damage. Yet, when your immune system is compromised by a poor diet, lack of exercise, smoking, excessive alcohol consumption, or chronic stress, these microscopic cancers can evade detection. Once they escape immune surveillance, they still encounter another crucial barrier. They require a blood supply to grow. A tumor that cannot access blood vessels is effectively frozen in size. It can grow to approximately 2 to 3 millimeters in diameter, roughly the size of a ballpoint pen's tip, and then its growth halts. Without oxygen and nutrients, it cannot expand further. Your body naturally endeavors to prevent blood vessels from reaching tumors through a process known as anti-angiogenesis. Angiogenesis, in essence, refers to the formation of new blood vessels. Your body meticulously regulates this process because a precise balance is required. Too few blood vessels can lead to tissue death from oxygen deprivation. Conversely, an overabundance of blood vessels can result in conditions like diabetic retinopathy, where excessive vessel growth in the eye leads to vision loss. Consider blood vessel growth like maintaining a lawn at the ideal height. Not too long, not too short, but just right. This is the Goldilocks zone your body strives to maintain. However, this is precisely what makes cancer so formidable. Cancer cells possess the ability to hijack your body's angiogenesis system. They release growth factors, which are proteins that act like fertilizer, stimulating new blood vessels to grow directly into the tumor. The moment these blood vessels connect, everything changes. Scientific research has meticulously quantified this phenomenon. In laboratory studies, when microscopic tumors are prevented from ever developing a blood supply, they remain harmless indefinitely. But when blood vessels are permitted to penetrate such a tumor, it can grow an astonishing 16,000 times larger in approximately 2 weeks. This represents explosive, exponential growth fueled by the readily available oxygen and nutrients. Understanding these biological mechanisms is paramount because it illuminates precisely where we can intervene. But this is only a part of the broader picture. One of the most groundbreaking advancements in cancer treatment over the past decade has been immunotherapy, which is revolutionizing the approach to treating colon cancer. Unlike chemotherapy, which directly destroys cancer cells, immunotherapy works by activating your own immune system to target and attack cancer. Cancer cells are remarkably adept at evading detection by the immune system, often employing strategies akin to wearing an invisibility cloak. They express specific proteins on their surface that effectively signal to immune cells, "I am normal, do not attack me." These proteins are referred to as checkpoints. Immunotherapy drugs, particularly checkpoint inhibitors, function by stripping away this cloak. They block these checkpoint proteins, thereby exposing the cancer cells and enabling your immune system to recognize and eliminate them. The outcomes can be nothing short of extraordinary. Patients diagnosed with stage 4 colon cancer, meaning the cancer has spread to distant organs, can achieve complete remission. This transformation from stage 4 to stage 0 is not universally experienced by everyone, and this variability is a significant focus of ongoing research. Nevertheless, when it proves effective, the results are truly life-changing. One of the most widely utilized checkpoint inhibitors is pembrolizumab, marketed under the brand name Keytruda. Other notable examples include nivolumab and ipilimumab. These medications are administered intravenously, typically every 3 weeks. They can be used as standalone treatments or in conjunction with chemotherapy, depending on the specific characteristics of the cancer. Now here's a critical detail. Not all colon cancers respond to immunotherapy. Your oncologist will conduct tests on your tumor tissue to identify specific markers that predict a response. A key marker is known as microsatellite instability, or MSI. Tumors exhibiting high microsatellite instability, MSIH, are significantly more likely to respond to checkpoint inhibitors. This is because MSIH tumors possess a greater number of mutations, making them more readily recognizable to the immune system once the checkpoint blockade is removed. Another important marker is PD-L1 expression. PD-L1 is one of those checkpoint proteins I mentioned earlier. If your tumor displays high levels of PD-L1 expression, immunotherapy drugs that target this protein are more likely to be effective. If you or someone you know has been diagnosed with colon cancer, it is imperative to inquire with your physician whether your tumor has been tested for MSI status and PD-L1 expression. These tests are instrumental in determining eligibility for immunotherapy, which can be life-saving. The second crucial mechanism, and arguably even more vital for understanding how nutrition can support cancer treatment, involves a groundbreaking study conducted in 2017 at the Gustave Roussy Cancer Center in France. This research involved 200 cancer patients undergoing immunotherapy. Some of these patients exhibited remarkable responses, with their tumors shrinking or completely disappearing. In contrast, other patients showed no response at all. The researchers were eager to understand the reason for this disparity. They meticulously analyzed every conceivable variable, including genetics, age, tumor type, and prior treatments. The sole significant difference identified between the responders and non-responders was the composition of their gut bacteria. Specifically, patients who responded positively to immunotherapy had elevated levels of a particular bacterium called Akkermansia mucinifila. Patients lacking Akkermansia did not experience a response. This pivotal study, published in the journal Science, fundamentally reshaped our understanding of cancer treatment. The significance of this finding lies in the role of Akkermansia. This is a beneficial bacterium residing in the mucus layer that lines your intestinal wall. It is considered a guardian bacterium because it reinforces your gut barrier, reduces inflammation, and crucially, primes your immune system to combat cancer more effectively. In experimental settings, when researchers transplanted Akkermansia from responding patients into laboratory animals that were not responding to immunotherapy, those animals suddenly began to respond to the treatment. The bacteria made the difference. Now consider the practical implications of this. If you are undergoing immunotherapy, the presence of a robust Akkermansia population in your gut could be the determining factor in whether the treatment saves your life or proves ineffective. Even for cancer prevention, maintaining healthy Akkermansia levels supports optimal immune function. The challenge, however, is that Akkermansia populations can be easily depleted. A single course of antibiotics, for instance, can reduce Akkermansia levels to near zero, and it can take weeks to months to rebuild these populations. So, how can you naturally cultivate Akkermansia? The answer lies in feeding it with specific foods. Akkermansia thrives on compounds known as elegotannins, which are found in foods like pomegranates, cranberries, and walnuts. Studies have demonstrated that consuming just 8 ounces of pomegranate juice daily can increase Akkermansia levels 6-fold within 3 weeks. Other foods that promote the growth of Akkermansia include oats and oatmeal, dried chili peppers, like the flakes often added to pizza, and Chinese black vinegar. You'll be interested to learn about the specific dietary protocol I've discovered that optimizes your gut bacteria for cancer prevention. The third pathway to consider involves directly starving tumors by inhibiting their blood supply through anti-angiogenesis. As previously mentioned, cutting off a tumor's blood supply effectively halts its growth. Pharmaceutical companies have invested billions in developing drugs designed to block tumor blood vessel formation. Medications like bevacizumab, marketed as Avastin, are now standard treatments for colorectal cancer and many other forms of cancer. These drugs function by inhibiting a protein called VEGF, vascular endothelial growth factor, which tumors release to stimulate the formation of new blood vessels. However, what has significantly surprised the research community is that when scientists test foods using the same laboratory assays employed in the development of anti-angiogenic drugs, many foods exhibit comparable blood vessel blocking activity. Green tea extract, for example, is so potent in its ability to inhibit abnormal blood vessel growth that if researchers were unaware of its plant origin, they would immediately advance it to clinical trials. The active compounds are catechins, particularly one known as EGCG. Other foods with powerful anti-angiogenic properties include tomatoes, which contain lycopene, cruciferous vegetables like broccoli and Brussels sprouts, which are rich in isothiocyanates, and sulforaphane, soy foods containing genistein, olive oil with its hydroxytyrosol content, and seafood abundant in omega-3 fatty acids. These effects are not insignificant. Population studies reveal that individuals who consume high amounts of these foods experience dramatically lower cancer rates. One study, which followed over 68,000 women for more than 10 years, found that those who drank green tea at least 3 times per week had a 27% lower risk of colorectal cancer. Daily green tea drinkers showed even more favorable outcomes. This represents a substantial reduction in risk achievable through a simple dietary habit. Now that you have a firm grasp of the underlying mechanisms of cancer prevention and treatment, the pressing question is, what concrete steps can you take? What are the specific foods and the exact quantities required? Allow me to present a precise protocol, grounded in research and aligned with my recommendations for patients at high risk for colon cancer. The absolute cornerstone of this protocol is dietary fiber. This is non-negotiable. Fiber serves to nourish your beneficial gut bacteria, reduce inflammation, accelerate transit time, thereby minimizing the contact of carcinogens with your colon lining, and directly supports immune function. A landmark study involving melanoma patients undergoing immunotherapy revealed that for every 5 to 6 grams of dietary fiber consumed daily, mortality decreased by 30%. For context, 5 to 6 grams of fiber is approximately the amount found in 1 medium pear. Current recommendations advocate for at least 25 to 30 grams of fiber daily for women and 30 to 38 grams for men. Unfortunately, the majority of Americans consume less than 15 grams. Therefore, it is imperative to double or even triple your fiber intake. The most potent sources of fiber are beans and legumes. Black beans, pinto beans, white beans, chickpeas, and lentils each provide approximately 15 grams of fiber per cooked cup. Aim to consume beans at least 4 times per week, ideally daily. 1 cup of cooked beans alone can fulfill half of your daily fiber requirement. If you are not accustomed to eating beans, it is advisable to begin gradually to avoid digestive discomfort. Start with half a cup 3 times a week and progressively increase your intake. Whole grains are another excellent source of fiber, including oatmeal, brown rice, quinoa, whole wheat, and barley, all of which contribute significantly to your daily fiber intake. 1 cup of cooked oatmeal, for instance, provides about 4 grams of fiber and also supports Akkermansia growth. Fruits and vegetables naturally contain fiber, but you will need to consume larger volumes to meet your target intake. Berries, apples, and pears are all excellent choices. Cruciferous vegetables such as broccoli, Brussels sprouts, and kale offer both fiber and potent cancer-fighting compounds. The second essential component of this dietary strategy involves polyphenols. These are plant-based compounds that nourish beneficial gut bacteria, reduce inflammation, and exert direct anti-cancer effects. The richest sources of polyphenols are deeply colored fruits and vegetables. Blueberries, strawberries, raspberries, and blackberries are all excellent sources, packed with anthocyanins. Aim for 1 to 2 cups of mixed berries daily. Pomegranates, whether consumed as fresh fruit or as 100% pure juice with no added sugar, provide L-glutanins that specifically promote the growth of Akkermansia. Consuming 4 to 8 ounces of pomegranate juice daily or half a fresh pomegranate is recommended. Green tea, brewed properly, is also highly beneficial. Aim for 3 cups daily. Ensure the water is heated to about 175 degrees Celsius, just below boiling, and steep for 3 to 5 minutes. Avoid adding dairy milk as its proteins can bind to the beneficial catechins. Adding lemon juice can enhance the absorption of these compounds. Coffee also contributes positively. Regular coffee consumption is associated with a lower risk of colorectal cancer. 2 to 3 cups daily provide chlorogenic acid and other beneficial compounds. Extra virgin olive oil is another key player. Consuming 2 to 4 tablespoons daily, whether on salads, vegetables, or even taken by the spoonful, is recommended. The polyphenols found in high-quality olive oil, such as hydroxytyrosol and oleocanthal, possess powerful anti-inflammatory and anti-angiogenic properties. Dark chocolate with a cacao content of 70% or higher, approximately 30 grams or 2 small squares daily, provides flavonoids that support vascular health and exhibit anti-cancer properties. The third component of this dietary approach is omega-3 fatty acids. Previously, it was believed that significant amounts could only be obtained from salmon and other fatty fish. However, research now indicates that even white fish like cod and halibut, as well as shellfish such as shrimp, mussels, and clams, contain beneficial omega-3s. These fats are inherently anti-inflammatory and anti-angiogenic. Aim for at least 3 servings of seafood per week, with a serving size of approximately 4 ounces cooked. If you follow a vegetarian or vegan diet, prioritize ground flaxseed, 2 to 4 tablespoons daily, chia seeds, and walnuts. Additionally, consider an algae-based omega-3 supplement that directly provides EPA and DHA. The fourth crucial component involves cruciferous vegetables. And here's a detail that many overlook. Broccoli sprouts are exponentially more potent than mature broccoli. Seedlings that are just 3 to 4 days old contain 100 times more sulforaphane than fully grown broccoli. Sulforaphane plays a vital role in activating your body's detoxification systems and possesses direct anti-cancer effects. A small handful of broccoli sprouts, roughly a quarter cup, consumed 3 to 4 times per week, can provide more benefit than eating pounds of mature broccoli. You can often find sprouts at grocery stores, or even grow them yourself in a jar within 4 days. If sprouts are unavailable, consuming regular broccoli is still beneficial. In this case, focus on the stems, which contain twice the amount of sulforaphane as the florets. Lightly steam cruciferous vegetables for 3 to 4 minutes to maximize nutrient bioavailability. Avoid boiling, as it can destroy sulforaphane. The fifth component includes soy foods. There is a persistent and inaccurate notion that soy is harmful due to its phytoestrogen content. This is fundamentally incorrect. The phytoestrogens present in soy, primarily genistein, actually block human estrogen receptors and offer protective effects against hormone-related cancers. Genistein is also recognized as one of the most potent anti-angiogenic compounds ever studied, capable of inhibiting tumor blood vessel growth by over 70% in experimental models. Population studies consistently demonstrate that a high intake of soy is associated with lower cancer rates. Aim for 2 to 3 servings of whole soy foods per week. Tofu, tempeh, edamame, and unsweetened soy milk are all excellent choices. It is important to avoid highly processed soy protein isolates and focus instead on whole food forms. Now, allow me to share some practical tips, drawing from my 15 years of clinical experience, for maximizing these protective effects. Timing is important. Your gut bacteria and immune system are most active at night while you sleep. Consuming your fiber-rich foods and polyphenols throughout the day ensures that these nutrients are available when these systems are performing their essential functions. It is also advisable to avoid large meals within 3 hours of bedtime, as active digestion can interfere with cellular repair and immune surveillance processes. Consistency is far more impactful than intensity. Eating a single cup of beans once a week will not yield significant benefits. However, consuming half a cup of beans 6 days a week can profoundly transform your gut microbiome. The same principle applies to green tea and berries. Consistent daily intake leads to sustained activation of protective pathways. Preparation methods can dramatically influence nutrient availability. Tomatoes should be cooked because heat is essential for releasing lycopene. Raw tomatoes provide very little lycopene. Tomato sauce, paste, and roasted tomatoes are ideal options. When cooking tomatoes, it's beneficial to add olive oil, as lycopene is fat-soluble. Cruciferous vegetables are best consumed lightly steamed or raw. Overcooking can diminish the beneficial sulforaphane content. For green tea, water temperature and steeping time are critical. Water that is too hot can degrade catechins, while water that is too cool may not extract them effectively. A temperature of 175 degrees Fahrenheit for 3 to 5 minutes is optimal. Variety is key to achieving synergy. Don't limit yourself to just blueberries. Rotate your berry intake among blueberries, strawberries, raspberries, and blackberries, as each contains unique anthocyanins that work together. Similarly, don't just consume black beans. Vary your intake with lentils and chickpeas. Different types of fiber support different bacteria, and the diversity of your diet directly influences the diversity of your gut microbiome, which in turn predicts health outcomes. If you are currently undergoing cancer treatment, these dietary strategies can enhance the effectiveness of your therapy. However, it is crucial to emphasize that this approach is not intended to replace medical care, but rather to optimize your body's response to treatment. Always communicate your dietary choices with your oncologist, as certain foods can interact with specific chemotherapy drugs. While most interactions are benign, it is essential to be informed. If your primary concern is cancer prevention, especially if you have a family history of the disease, these foods should form the foundation of your daily diet. Perfection is not the goal. Consistent consumption of high-impact foods is. Aim for beans or legumes daily, berries daily, green tea or coffee daily, cruciferous vegetables at least 4 times per week, seafood at least 3 times per week, and extra virgin olive oil daily. This comprehensive approach covers the essential bases. A common mistake I observe is the belief that one needs to overhaul their entire lifestyle overnight. Instead, start with a single, manageable change. For instance, add a cup of beans to your lunch or dinner and maintain this consistently for 2 weeks. Then, incorporate a cup of berries into your breakfast. Continue this gradual approach for another 2 weeks. Subsequently, add 3 cups of green tea. Building these habits incrementally leads to sustainable change. Another mistake is assuming that supplements can fully replace whole foods. While fiber supplements Omega-3 capsules and polyphenol extracts can have a role, particularly if access to certain whole foods is limited. They provide isolated compounds. Whole foods, on the other hand, offer hundreds of compounds that work synergistically. A cup of blueberries, for example, contains not only anthocyanins, but also vitamin C, vitamin K, manganese, fiber, and numerous other polyphenols that enhance each other's effects. This synergistic interaction cannot be replicated in a capsule. Whole foods are invariably more potent. One final critical point: if you experience any digestive symptoms, such as blood in your stool, persistent changes in bowel habits, or unexplained weight loss, please get screened. Do not delay. Colon cancer is highly treatable when detected early, but can be deadly if diagnosed late. Current screening guidelines recommend colonoscopies starting at age 45 for individuals at average risk. If you have risk factors or are experiencing symptoms, do not wait until you reach the recommended age. Consult your doctor immediately. Ultimately, colon cancer does not have to be an inevitable consequence of aging, and stage 4 disease does not have to be a death sentence. The patients I encounter who successfully prevent cancer or achieve remission from advanced disease are not genetically superior or exceptionally lucky. They consistently support their body's natural defense systems through informed nutrition. Your immune system is capable of recognizing and eliminating cancer cells if it is provided with the necessary support. Your body can prevent tumors from developing blood vessels if you consume foods that activate anti-angiogenic pathways. Your gut bacteria can prime your immune system to respond effectively to treatment if you nourish those bacteria appropriately. These remarkable systems are inherent within you. The crucial question is whether your daily choices are activating them or suppressing them. In summary, we have explored how rising colon cancer rates in younger individuals are primarily attributed to diet and lifestyle factors, including processed meat consumption, sedentary behavior, and poor gut health. Your immune system is constantly at work eliminating microscopic cancers, but it requires optimal support to function effectively. Tumors are fundamentally incapable of growth without access to blood vessels, and certain foods can inhibit tumor angiogenesis with a potency comparable to some pharmaceutical drugs. Immunotherapy has the remarkable ability to reverse even stage 4 cancer by invigorating your immune system, but its success is heavily reliant on the composition of your gut bacteria. Akkermansia mucinifera is a key bacterium that significantly influences immunotherapy response and can be cultivated through the consumption of foods like pomegranates, cranberries, and oats. The core dietary protocol includes a daily intake of 25 to 38 grams of fiber from sources such as beans, legumes, and whole grains, 1 to 2 cups of berries daily, 3 cups of green tea, or 2 to 3 cups of coffee daily, at least 4 servings of cruciferous vegetables weekly, with a particular emphasis on broccoli sprouts, 3 servings of seafood weekly, 2 to 4 tablespoons of extra virgin olive oil daily, and 2 to 3 servings of whole soy foods weekly. Consistency and appropriate preparation methods are far more impactful than striving for absolute perfection. Your genetic predisposition does not predetermine your cancer risk. Your daily dietary choices exert a profound influence. If you found this information valuable, please consider subscribing and liking this video. Your engagement helps me reach more individuals seeking evidence-based insights into disease prevention and treatment support.